Okay, I'm live. Okay. So, my dear student, okay, yes, Baisakhi, happy Baisakhi, ma'am. Yeah, sure. So, good evening, everyone, those who are live. So, today is a very important topic, and I would like to tell that nobody should miss this topic. Actually, why it is very important, I will tell you, because for proper understanding of neuroanatomy, in many of the colleges, yeah, I am good. I am good, dear. So in many of the colleges already the neuroanatomy has finished, and for the proper understanding of neuroanatomy, it is important that we should be knowing about the general aspect or the general anatomy aspect of the general aspects of the nervous system. That is very important. How it is classified as the autonomic nervous system, as the central nervous system, what is neuron, what is cell body, what is neuroglial cell. So these are certain concepts which comes from the general anatomy sessions only. So actually today is the last session of general anatomy series. Already those who have missed out, I have covered the first topic of general anatomy, that was planes, and about all the important bony structures and different kind of planes. Then I have talked about skin, fascia, deep fascia. I have also talked about the parts of the muscles variety, the bones, the cartilages, the joints and their subtypes. And today is the neuroanatomy basis, that is the introduction to nervous system. For understanding of neuroanatomy, we have to understand general anatomy of nervous system. Okay, so let me start with the session. So first thing is that nervous system is a very important topic in which we will study about the neurons. What is neurons? Neurons is the anatomical and functional unit of nervous system. Yeah, hello dear. So please let's write it. Okay, before that, I will write the important aspect. Before that, I want everybody to see this chart. So when I'm talking about nervous system, it can be categorized as central nervous system and peripheral nervous system. So if I'm talking about central nervous system, it is consist of brain and the spinal cord, the central nervous system part. And if I am talking about the peripheral nervous system, it is consist of all the structures which is lying, all the sensory structures which is lying outside the brain and the spinal cord. It can be categorized as the somatic and autonomic system. Okay, nervous system. So when I talk about somatic nervous system, the function of the somatic nervous system is to control voluntary activity of skeleton muscle. That means somatic nervous system is under our control. Okay, we can control it. Okay, if I want to tell you to flex your elbow, extend the elbow, this action is under somatic innervation. Okay, so the action which is controlled by somatic nerves are very important, and they are actually somatic nerves will give innervation to skeleton structures. So what about the autonomic nervous system? The autonomic nervous system is further categorized as, okay, so if I am talking about autonomic nervous system, it is categorized as sympathetic and parasympathetic type. The sympathetic nervous system works in fight or flight stage, and the parasympathetic nervous system works in the rest and digest state. So this is important for you to understand. Now, I would like everybody to know that neurons, if I am talking about nervous tissues, it is consist of neurons and neuroglial cells, which is supporting the neurons. And if I am talking about the neurons, they are anatomical and functional unit of nervous system. The neurons is specialized structure which is consist of cell body and its process. Okay, so all the points which I am explaining, always have a notepad with you. Either you can write in your iPad or you can write in your notes the important points about the nervous system. It's very important, and I have found many students having difficulty for the basic understanding of nervous system. So this class is very important. So before proceeding into the structure of neurons, so the next the topic which I want everybody to know is the structure of nervous system. I want you to note down few important topics, few important things about introduction part. So if I am talking about introduction part, please note down this. Nervous. So the important point is, if I am talking about nervous system or nervous tissue, it is consist of neurons and neuroglial cells. Okay, that is clear. Other is neurons. What is neurons? So it is also important for you to know what is exactly neurons. So let's talk about neurons. Neurons are specialized cells which is consist of cell body and processes. It is consist of two important parts: cell body and its process. And the neurons, if I'm talking about the neurons, they are fundamental structural and anatomical unit of nervous system. So neurons are the unit of nervous system. So neurons are anatomical and functional unit of neurons are anatomical and functional unit of nervous system. Okay. Now next thing which everybody should know is about the structure of the neurons. So we will firstly talk about, so if I have to give you an elaborate description of the structures of neuron, I would like firstly to explain about the cell body and then the processes, that is dendrites and axons. So let's focus firstly on the topic of structure of neuron. So when I'm talking about the structures of neuron, what all things you have to know? So the first thing which I want everybody to know is about the structures, that is the cell body. So firstly I'm going into the cell body description, then I will go into the description of dendrites and axons. Okay, so let's talk about the cell body. So first thing is that let's see this image in which we can see the structure of neuron, in which we can see the cell body of neurons. Now, what is the other name of the cell body of neuron? It is also called as soma. So, the cell body of neuron is also called as soma or pericrion, and it is actually, as you can see, it is actually in most of the part, if I am talking about the part of neurons, where is the cell body located? So, the cell body of the neurons are mainly located in the gray matter of brain and spinal cord. So they are actually located in the, so cell bodies of the neurons will be present in the gray matter, and white matter will be present, will contain nerve fibers. Okay, so let's, if I want to tell you about the size variation, the size variation of soma or cell body is varying from 5 to 100 micrometers diameters. Okay, so that is important for you to note down. And also we can see the processes. So one by one I will tell about each and every aspect. So first let's write important point. Cell body, which is also called as soma. Okay, all the names you have to know. What is the other name? It is also called as pericrion. And most of the, if I am talking about most common location of the cell body or the soma, where it is found, so where it is located, so it is located in the gray matter of brain and spinal cord. Okay, it is located in the gray matter of brain and spinal cord. Now, what is the average dimension or variation of the dimension? Is 5 to 100 micrometers in diameter. Now, let's talk about the features of cell body. So this is actually the image which is shown you on the other hand, is showing the structure of the cell body. And if I'm talking about the structure of cell body, what all things you can see? Inside the cell body, we can appreciate here, there is nucleus which contains nucleolus, as you can see in the diagram. Okay, so we will have exactly the nucleus which is placed centrally, and it can also placed eccentric in location when there is nerve injury or the case of fatigue of neurons, or in the sympathetic ganglia it can be eccentric. Inside the nucleus we have got nucleolus. And thus, if I talk about important structures, so cytoplasm, if I am talking about the cytoplasm of the cell body, inside there is Nissl granules. So you can see there is Nissl granules, Nissl bodies. So these Nissl granules and the overall, if appearance ki baat karte hain, if we are talking about the appearance of the cytoplasm part of the cell body, it is basophilic. It contains rough endoplasmic reticulum and it contains ribosome, and it is very important that also, if I am talking about cell body, it is having a trilaminar structures. It is having a bilayered protein, and intermediate layer is made of lipid layer. Okay, so it is trilaminar. That is also very important. So let's write few important points about the cell bodies. Okay. Okay, so cell body, if I am talking about the cell body part, it is dimension we have written. It is covered by a cell membrane. So the covering of the cell body is called as cell membrane. Okay, which is trilaminar, which is having tri-layered. And when I am using the word trilaminar, what it means, it is consist of bilayered, it is consist of bilayered protein. Bilayered protein means the protein layer will be inner and outer aspects. This protein layer will lie inner and outer, and in between the inner and outer protein layer, intermediate layer will consist of lipid layer. The intermediate layer is containing lipids. Okay, now, so this is an important point, and the nucleus is next important point which I want you to tell about this structure, that it contains inside nucleus. It contains nucleus. The location of nucleus is in most of the cases it is centrally placed, and it is having a prominent nucleolus. It is having a prominent nucleolus. Okay, okay, done. Okay, now, but sometimes if I talk about the nucleus, its location is eccentric. So sometimes the location can be eccentric. And when is the location eccentric? When is the nucleus location eccentric? It is eccentric in the case of injury, if there is an injury to neuron, if there is a case of fatigue, when the neurons has become fatigue. So fatigue of neuron, when the case is of injury of neuron or the neurons has weakened, fatigue, or normally also it can found in the cases of sympathetic ganglia. Okay. So these are the features or the structural features which we are seeing. These are the structures features which we are seeing for the cell body. Okay. Now, inside the cell body, what I have told you, also there is presence of Nissl granules. So let's talk about that. So again, I would like to show you this image. So this is another view in which you can appreciate few important points about the Nissl granules. Also, you can see the cytoplasm. The cytoplasm, if I am talking about the cytoplasm of the cell body, this is the cell body which is containing inside the cytoplasm. And if you will see the cytoplasm, it has got an appearance of basophilic appearance, which will also contains rough endoplasmic reticulum, ribosome, and that is important for protein synthesis. Okay, so that is an important point which you have to know. And let's write cytoplasm of the cell body contains Nissl granules. It also contains, the appearance is basophilic, and it contains rough endoplasmic reticulum containing ribosomes, important for protein synthesis. Okay, this is, and other than that, so actually one more thing which I want you to appreciate, can you see these are all Nissl granules? Okay, so what you can see, the Nissl granules which you are seeing, and also you can see this is the nucleus which is located here in the cell body center, and these Nissl granules you can see are extending into dendrites also, but the Nissl granules are absent in the axon and axon hillock. This part is called as axon hillock. Okay, So basically there is absence of nacelle granules in the axon and axon hillock part, but the nacelle granules can extend into dendrites. This is an important point which everybody should note down. So I will write in short nacelle granules as NG. Is it okay? What is NG? NG means nacelle granules. Okay. Nacelle granules can extend, so it extends into dendrites. It extends into dendrites, but absent in axon hillock and axons. Okay. The nacelle granules are active in protein synthesis. So what is its function? Nacelle granules are active. They are active in protein synthesis. Okay. Now, other than that, other structures which will lying in the cytoplasm is the Golgi bodies, which is lying close to the nucleus, and the mitochondria will be obviously present. So you can see this image where you can see the presence of nacelle granules are there. Other structures which are present are mitochondria, the Golgi apparatus structures, microtubules has been also seen extending in the axon. So more other structures which has been seen here, Golgi bodies located close to the nucleus. Also, it contains mitochondria. Okay, also it contains mitochondria. Now, other than that, there is also the presence of lysozymes inside. Okay, so we will have the membranous vesicle which is residing in the cell body, that is called as lysozyme. So, also there will be lysosomes that is called as membranous vesicle. What is it called as? Membranous vesicle. And what are lysosomes? So they are hydrolytic enzyme. So basically they will secrete, this lysosomes, which is membrane bound vesicles, they will secrete hydrolytic enzymes and acid phosphatase. So they will secrete hydrolytic enzymes and hydrolytic enzymes and acid phosphatase. Okay. Now, actually, this is an important point. So, if I talk about the function of lysosomes has got a function of phagocytizing foreign body and hydrolyze the nacelle granules and during the process of chromatolysis. So, this is an important function of lysosomes. So, please write down lysosomes phagocytizes the foreign body, FB for foreign body, and hydrolyzes the nacelle granules. Okay, during the process of chromatolysis. Okay, now, so this will also have the, so basically, if I am talking about aged neurons, there is also present of lipofuscin, which is actually also called as wear and tear pigment. So pigment is also seen in the case of which is called wear and tear pigments, which is golden brown in color and seen in the cell bodies of aged neurons. So this is an important point which you have to note down. Please note down lipofuscin. So I just want everybody to mark it as a note point. This is the pigment which will be seen in the cell body, and this pigment is golden brown in color. What is the color? The color is golden brown. My dear students, are you understanding? Please let me know. Any confusion? You just let me know. These are the basic things which you should know. Also, the details will be done in the histology part. Okay. So, today I am talking about the general aspect of the neurons, the general structures. But when we take a session of histology, more of details and histological images will be shown to you. Golden brown pigment seen in the cell bodies of aged neurons. Okay. So, and the other important point is, so we can also define lipofuscin, which was present in the aged neurons, as it is a wear and tear pigment. What is it? It is a wear and tear pigment. It is wear and tear pigment. Done? Okay. So, my dear students, are you understanding? Let me just check your messages, everyone. Yeah, hello everyone. If any of the students are not understanding anything, please let me know. I will explain everything once again. So what till now, what all things we have covered? We have covered the structures of the cell body part of the neuron, where we have covered and we have seen that neurons are trilamellar structure, having bilayered protein and intermediate layer made of lipid. And that means it is having insulations. And also we have talked about that it is having nissl granules inside. It is having cytoplasm containing the nissl granules. It is also having mitochondria, Golgi body, location of nucleus. And when the location of nucleus will be eccentric in the case of if there is an injury to the neurons, if there is weakness of the neurons, and that all we have done. And also in the sympathetic ganglion, which we have seen. Okay, other than that, which we have already seen. And a very important point, which is very important for MCQs, please note down, this is important for MCQ and the Viva, that if I am talking about the cytoplasm, it contains nissl granules, but nissl granules will be present in the cell body. It can extend into dendrite, but it will not extend into the tapering part or the extension, that is axon or axon hillock part of the neurons. Also, it is actually very important. Nissl granules important for protein synthesis. Also, we have talked about Golgi bodies, location, lysosomes, which is very important, membrane vesicles, which has got hydrolytic enzymes and acid phosphatase. And we have also talked about the lipofuscin. So done with this. Now let's talk about the difference between. So this concept is also important. Many times I have seen the students and I have asked with the students about the concept which they are knowing about the difference between nuclei and ganglia, nucleus and ganglia. So it is very important that you should be aware of this difference. Okay. Okay, dear. Till now, is it okay? So let's talk about. So it is important that we should be knowing that collection. If I am talking about nucleus, the collection of the cell bodies, nerve cell bodies, which is lying within the central nervous system, is nuclei, and the collection of nerve cell bodies which is lying outside the central nervous system is called as ganglia. So this is the basic difference between nuclei and ganglia. Okay, these are the basic concepts which can be asked when in the main examination, in university examination, also during the Viva. So please write down the difference. Okay, so write down the difference. Let's start writing it. Okay, so we will write nuclei. And we will write about ganglia. Okay. Okay. So collection of nerve cell body within the central nervous system is nuclei. Collection of nerve cell body, collection of nerve cell bodies within central nervous system is called as nuclei. And collection of nerve cell bodies outside central nervous system, outside the central nervous system, is called as ganglia. Okay, now it's also important for you to know, that means examples of ganglia will be, so it is not residing inside the central nervous system. Example is dorsal root ganglia of the spinal nerve and the ganglia which is associated with cranial nerve. So these are two examples. These are the examples where we have got the cell bodies, the nerve cell bodies lying outside the central nervous system. Two examples, my dear students, please note down. That is dorsal root ganglia of spinal nerves, and the second is also the ganglia in relation to cranial nerves. Done? Okay. So that means we have got the concept of nuclei, we have got the concept of ganglia, and we are knowing the ganglia's example also, dorsal root ganglia and the ganglia associated with cranial nerves. Okay. Now let's talk about more. So I would like to now talk about the process, that is dendrites. Okay. So in this diagram, we can see the axon. We have also discussed the axon. Now we are focusing on the dendrites. So we can see short multiple processes arising from the cell body. So in this diagram, if you are just if I'm just enlarging this image, okay. So if I'm enlarging this image, please see this. This is cell body containing N for nucleus, which we have discussed. Inside there is lipofuscin pigment. Inside there will be nissl granules. Inside there will be mitochondrias, Golgi bodies, rough endoplasmic reticulum. Okay. So all this we have done. Now, if I am talking about these structure, that is the projections, we can see multiple processes arising from the cell body extending a branching pattern which is called as dendritic tree. Okay. So this is important for you to understand. So please write down about dendrites. So let's focus on the dendrites. So what is dendrite? Dendrites are short multiple processes arising from cell body, showing extensive those dendrites showing extensive branching form, they are called as dendritic tree. What they are called as? They are called as dendritic tree. Okay. Now, it's very important that you should also know that dendrites will contain all cytoplasmic contents which we have discussed in the cell body. We have discussed in the cell body so many of the cytoplasmic contents. So the content will be also present in the dendrites. So write down. Dendrites contains all cytoplasmic content same as cell body. So no need to write everything once more. You just understand by one line that the content will be same. The dendrites will also have the content as same as the cell body. So other than that, it is also important for you to know that we are talking about dendrites. So if we are talking about dendrites in central nervous system, they are providing structural support for the neurons. So this is an important function. And if I am talking about peripheral nervous system, they also contribute in formation of myelin sheath for the axons, and they are also nutritional. Okay. So they are actually, dendrites are capable of mitotic division throughout their life. So these are certain points which you should know about dendrite. So let's write it. Then I will show you one more diagram. Okay. So let's write few more important points of the dendrites. Dendrites in central nervous system, they provide structural support for neurons. So an important function is giving support to the neuron. Okay. Now, so this is in central nervous system. What about their role in peripheral nervous system? So when I am talking about peripheral nervous system, the other function is that they are also forming, they form myelin sheath. They contribute formation of myelin sheath for axons, and they are also nutritional. They have also got nutritional function to perform. They are also capable of mitotic division throughout the life. This is another important point. Note point about dendrites. They are capable of mitotic division throughout their life. Okay, done. So now let's see one image here. So once more, I would like to show you this image. So this is an image where you can see the cell body of the neuron. You can see the nucleus location. You can see rough endoplasmic reticulum, Golgi apparatus residing inside, the mitochondria, the dendritic branch, dendritic tree, axon hillock, and the axons. Okay. So let's talk. The next thing is to know, let's talk. So we have known, we are now knowing about the cell body of the neuron. We are now knowing also about the dendrite part of the neuron. Now we have to discuss about the axon hillock and that of axon. So we will discuss about the axon of the neuron. So let's see here. So basically, can you see the long process? If I am talking about the neurons, which is containing cell body and dendrite, there is long process. Can you see the long process which is projecting? That is called as an axon. So each neuron can have having a single axon and a long process which is variable in length. The length of axon is variable. So that is an important point. As you can see, so this is the axon. Okay, these are the axons which is shown in the image. Okay, now axons. Each neurons has single. So this is single. Each neurons has got single axon and this is a long process. So if I am talking about axon, it is the long process of neuron and is a long process. It's a long process variable in length. Long process variable in length. Okay, it's a variable in length. So basically also it is important that the area where axons, so where the axons are arising. So origin part of the axon. So we have got the cell body, we have got the projection, which is multiple branching pattern, which is seen from the cell body. What is it? It is the dendrite, which I have talked, and it has a tapering end. It has got a long process, which is axon, and the origin of the axon is showing a pyramidal structure close to the cell body, that is called as axon hillock. Okay, so what is axon hillock? Is close to the origin of, it is close to the origin of axon, which is actually a short pyramidal structure. It's a short pyramidal structure. So basically, actually, if I talk about one more important point, which you already know, both axon and axon hillock is not having nissel granules. So they have got no nissel granules. A note point, which I am again repeating because this will be asked in your Viva examination. So please write down axon and axon hillock. Is not having, it is not having nissel granules. It is not having nissel granules or substance. Okay, done. So yes, we have done with this. Now let me show you image once more. Okay, so here you can see this area is the area of axon hillock. Okay, this pyramidal structure which you can see is the axon hillock, which is close to the it is actually pyramidal structure near the origin part where the axons are originating. Okay, now if there is enveloping structures around the axon, so that is called that is actually enveloping cells are Schwann cells. So I would also like to show you that. So let's see here. So see here. So what we can see in the case of axon, what can you see? There is a sheath, axon membrane, and there is sheath which is lying outside. The squam cells are there, so also we can see the squam cells residing outside. Okay, this is the squam cell. So basically, actually axons have concentric lamella, so concentric wrapping is there enveloping, and this is the concentric wrapping, as you can see, in the form of myelin sheath. The axon cells forms the myelin sheath in peripheral nervous system. And if I talk about oligodendrocyte cells, they form the myelin sheath in case of central nervous system. So this is again very, very important for you to note down. Okay, so basically this is an important point, so please note down. Note. Okay. Okay, dear. So thick axons have concentric wrapping. They have concentric wrapping enveloping them, which is actually forming the myelin sheath by squam cells in case of peripheral nervous system. Okay, that is shown here. Here in this diagram, you can also see the nucleus of squam cells here, and this myelin sheath, which is a wrapping or insulation, which is seen here around the axon, is called as a myelin sheath, which is formed by the squam cells in case of peripheral nervous system, and it is formed by oligodendrocytes in case of central nervous system. Now, again note point. Oligodendrocytes in case of central nervous system forms the myelin sheath. Actually, these two MCQs are often asked in examination, that which cells forms myelin sheath in central nervous system enveloping the axon, and which structure forms the myelin sheath in case of peripheral nervous system. So in peripheral nervous system, that is the squam cells which forms the myelin sheath, and in central nervous system, the oligodendrocyte cells are forming the myelin sheath. Okay, so now see here one more image. So basically you can see the axon, you can see the squam cell nucleus as in this diagram, and you can see this is the this is exactly the myelin sheath. And also I want you to add on one more point, actually, that is also very, very important for you, that outside the myelin sheath, there is a large layer of squam cells, and its cell membrane is again forming an additional sheath, which is called as neurolimal sheath. So we can also see the neurolimal sheath here. This is the neurolimal sheath. Okay, so that is important. And this neurolimal sheath is giving additional protection, and it is helping in the process of regeneration of the peripheral nerve in case of injury happening to peripheral nervous system. Okay, so as you can see in this diagram, there is myelin sheath, and outside there is also neurolimal sheath. Okay, so that is seen here. So there are multiple myelin sheath, and outside that we have also got a neurolimal sheath. So please write down this. Outside myelin sheath, in peripheral nervous system, we are talking about peripheral nervous system. In the case of peripheral nervous system, there is thin layer of squam cells. There is thin layer of squam cells and its cell membrane, forms an additional shell sheath called as neurolimal sheath. Neurolimal sheath. And what is the function of this neuroglial sheath? So important to know the function. So let's write neuroglial sheath helps in regeneration of peripheral nerves. Okay, after injury, that is in case of peripheral nervous system. Okay, this is the function. This is an important function. Okay, now again I would like to show you this image where you can see the axon hillock area. You can see the axons. Okay, so if I am talking about important point about the axon, I would also like to show you that actually there is a sometimes what happened, the axon is covered by myelin sheath, but there is a gap where there is absence of myelin sheath, that is called as knots of Ranvier. So let me show you an image where you can see the knots of Ranvier. Yeah, can you see here? In this image, you can see there is axon, and the axon is there is axon which is covered by myelin sheath here and here. So there is a gap in between. So there lies a gap in between this area where we have got no myelin sheath. So that area is called as knots of Ranvier. Okay, knots of Ranvier. So please write down this. That is important. Knots of Ranvier is a gap in axon where they are not covered by myelin sheath. Okay, they are not covered by myelin sheath. Okay, so basically this is important. And if I am talking about the area between the knots of Ranvier, that part is called as that area is called as internode. So each unit of axon which is lying between the two knots of Ranvier is called as internode. So that is again important point. So write down each unit of axon between two knots of Ranvier, if I am talking, is called as internode. What is it called as? It is called as internode. Okay. Now, if I am talking about the termination of axon, it happens in the form of telodendria. So let's see the telodendrias. See here. Can you see here? The termination of axons is in the form of telodendria. So that is important that you should note down. Okay. So what is telodendrias? Before classification of neurons is the next type. So what is it? Please write down as a note point. Okay. That is the termination which happens in the, so near the termination of axon dendrites are there, and these axon dendrites are termed as telodendria. What is it called as? Telodendria. Okay, this is called as telodendria. Now we have done about the general. So now we have talked about much about the general features of neurons. We have talked about the cell body. We have talked about its, we have also talked about the axon hillock. We have talked about dendrites. Next is the classification of neurons. So the classification of neurons is actually, it can be classified as the various types according to different types of classification. So according to the polarity, the classification of neurons can be like, it can be unipolar, it can be bipolar, it can be pseudo unipolar, it can be multipolar. Okay, so first I want you to show this image. Okay, so you can see this image and you can appreciate that basically we have got following types of neurons. So here we can see we will have the unipolar neurons, we will have the bipolar neurons, we will have pseudo unipolar neurons and multipolar neurons as shown in this image. Okay, so this is the classification which is according to the polarity. This is the classification according to the polarity. So if I am talking about unipolar neurons, these are having one process, as you can see here. Okay, so this is extending from, so we will have one process which is extending, as you can see, from the cell body. So then I will also give you the example of each variety. So basically example of unipolar neurons is mesencephalic nucleus of trigeminal. Now in the second image, you can see the bipolar neurons, which is having two processes, as you can see here. So neurons have got dendrites and axons, two process examples of neurons of retina, olfactory neurons, neurons of the vestibular cochlear nerve. These are all examples of bipolar neuron. Pseudo unipolar, as you can see, this is false unipolar as it is having a single process, which is now this single process is bifurcating. Can you see here? This is single process is again bifurcating as in this image. So this single process is bifurcating, and this kind of neuron is called as pseudo unipolar neurons as shown in this image. So which type of neuron is this? This is pseudo unipolar neurons. Example is dorsal root ganglia. Okay, of the spinal nerves. Multipolar, the most common is the multipolar type, in which we have studied the structure also. It will have multiple dendrites, one axon process, and will have cell body. And basically the neurons of spinal cord, the neurons of cerebrum, brain, cerebellum, these are autonomic ganglia. All these are example of multipolar neurons. So this is how we can define unipolar, bipolar, pseudo unipolar, and multipolar neuron. So let's write it. Okay, so firstly we will write about, so on the basis of polarity we have got following types of neurons. So the first type which I want to explain is unipolar. Actually, why it is important? Because so many times in the examination, the question has been asked about each variety. Okay, so the question has been asked multiple times in the Viva, and also in the university papers. It can be asked for the MCQs, or it can be asked for an enumeration question. So unipolar neurons have single process that is extending from cell bodies, that is extending from cell body, basically the dendrites. Examples. So what is the example of unipolar neuron is mesencephalic nucleus of trigeminal nerve. Mesencephalic nucleus of trigeminal nerve. Okay, so basically जब हम trigeminal nerve पढ़ाई करेंगे, when we will study about the trigeminal nerve, we will know that trigeminal nerve is a mixed type of nerve having motor component and having sensory component. The sensory component is again having nucleuses of three varieties. Spinal nucleuses, it will have the chief sensory nucleus and mesencephalic nucleus, which is important for the sensation of, which is important to perceive the proprioceptive sensation. And the mesencephalic nucleus of trigeminal nerve will extend into the midbrain part of the brainstem. Now coming to the second type of neurons described on the basis of polarity, that is the bipolar neurons. Bipolar neurons. Okay, dear? Bipolar neurons. So bipolar neurons means it is having two process. Have two process. So basically it is having two process. Okay, one dendrite and one axon. What is it? One dendrite and one axon. One dendrite and one axon. and the examples. Let's write the examples. So let's write the examples of bipolar neurons. So bipolar neurons examples are, it is present in retina, it is present in olfactory epithelium, okay, it is present in the sensory ganglia of vestibular and cochlear nerve, sensory ganglia of vestibular and cochlear nerve. Okay, so done. Now, which is the third category which we have seen? That is pseudo-unipolar neurons. So let's write about pseudo-unipolar neurons. Third category is pseudo-unipolar neurons. So let's write about pseudo-unipolar neurons. Okay, dear. So if I'm talking about pseudo-unipolar neurons, is having a single process, and this single process will expand, will extend, and then bifurcate. Okay, so it is called as false unipolar. So have single short, it is having single short process that extend from cell body and bifurcate. What is it? T shape. So you will see it in the form of T shape. Okay, so now let's talk about the example. So the example is sensory ganglia. So the example is sensory ganglia, and the name will be, so it's also it's present in dorsal root ganglia of spinal nerve. Sensory ganglia, dorsal root ganglia of spinal nerves. Okay, now coming to the most common type, the last type, which is divided according to the basis of polarity, is the multipolar neurons. Let's talk about that. The fourth type, multipolar neurons. Okay, multipolar neurons. So multipolar neurons will have single axon, will have cell body and several dendrites. Will have cell body, single axon, and several dendrites. Now it's important to know the example of multipolar neurons. Examples of multipolar neurons. The examples are the neurons which is present in, so the neurons which is present in spinal cord, which is the most common, is the multipolar neurons. It is present in the neurons of spinal cord. It is present in the neurons of cerebrum, cerebral cortex, cerebellum, and also autonomic ganglia. Okay, so done. So we have now defined the various variety of neurons, which is actually we have got 4 types according to the polarity. We have discussed unipolar, bipolar, multipolar, and pseudo-unipolar. Now we will discuss, and we will take the second category and we will discuss it. Okay. Yes, good evening, Mudit Gupta. Yeah, good evening, dear. Yes, this is free. I am preparing for, yes. So all of you can have the basic knowledge of neurons. This is highly beneficial. So study is always useful. So don't worry. Yes, neurons and its basic knowledge is very, very important. Okay, now let's talk about the classification of neurons, which happens according to the length of processes. So according to the length of process, we can classify the neurons as the Golgi type 1 variety and Golgi type 2 variety. So it is according to the length of process, mainly the axon. So it is according, we will define the neurons as Golgi type 1 and Golgi type 2 variety, which is according to the length of long process axon. So according to the length of process, the neurons will be classified as Golgi type 1 and Golgi type 2. Okay. Okay. Now, if I am talking about type 1 variety, it has got long processes like axons, and it will have short process. Now, if I am talking about the important example, so long process Golgi type 1 variety, it is present in the pyramidal cells. It will be present in the pyramidal cells of the cerebral cortex, and it will be present in the Purkinje cells that is present in cerebellum cortex. Okay, so these are two examples. So please write down. Examples are pyramidal cells of cerebral cortex, pyramidal cells of cerebral cortex, and Purkinje cells of cerebellar cortex. Okay. Now, Golgi type 2 has got short process, and it is present in, it is also short process cells, and it is type 2 variety is also present in the case of cerebellar cortex, cerebellum cortex neurons. Okay, so this is major variety. Now here you can see, here I would like to show you the diagram where you can see important pyramidal cells. So as there was example of long process, the pyramidal cells, so we can see the pyramidal cells. Can you see here? These are all pyramidal cells. These are all pyramidal cells of the cerebral cortex. Enlarged view has also been shown. These are all pyramidal cells. I would like to show you a history. So pyramidal cells are present in the cerebral cortex, and I would like to show you an histological image where you can identify, and you can see this pyramidal shape structure which is having neurons here. These are present in cerebral cortex, and this will be the example of Golgi type 1 variety of cells in the cerebral cortex. Actually, when we will talk about the neurons, when we will talk about the class of cerebral cortex and cerebellum, I will explain each of the histology part also there. Now here we can see the cerebellar cortex, in which we can see here that it is having Purkinje cells. Can you see flask shape cells are shown here, that is in the Purkinje cell layer. Now that if we talk about cerebellum, from outside to inside, the cerebellum is 3 layer. Cerebellar cortex is 6 layer, which was seen before, that was containing pyramidal cells. But in the cerebellum we have got outer molecular layer, intermediate Purkinje cell layer, and we have got inner granule cell layer. Okay, so one more histological image I would like to show. Actually, this is the histological image in which you can see the you can see the low power view and the high. So this is showing you the low power view, which is showing all the 3 layers: the molecular, the outer layer, then we have got the ganglionic layer, that is Purkinje cell layer, and granular layers. So in the high power view, if you will see, if just enlarge this, if I am enlarging this, can everybody appreciate? So can everybody here can appreciate the Purkinje cell layer of the cerebellum? So these cells, which is flag shape, can you see the flag-shaped cells? This has also been asked for an image-based question in various competitive exams. This flag-shaped cells are Purkinje cells. Okay, so done. Now we have done this. Now we are knowing that according to the length of process, we have defined the neurons into Golgi type 1 and Golgi type 2 variety. We have also defined according to polarity, and now I would like to define the neurons according to the shape of cell body. Okay, so if I am talking about the shape of cell body, we have got various types of neurons. We will have, according to the shape, we will have following types. We will have stellate, satellite or stellate cells. We will have flask shape cell. We will have pyramidal cells. Now, flag-shaped cells are present if I am talking about flag-shaped cells, we have seen, I have shown you in the high power view also, it is Purkinje Purkinje cells of cerebellum cortex. Where it is present? It is present in the cerebellum cortex. And pyramidal cells, that also I have shown you in large view. It is present in the cerebellar cortex. It is present in the cerebellar cortex. Okay. Now let's see also. So, and if I'm talking about stellate cells, it is present in the spinal cord. So see this diagram. Okay. So I will show you various diagrams here. Here in this diagram, we can see the stellate cells, which is basically, you can see here, this is star shaped cells like this. This is stellate cell. So this is the stellate cell, and it is present where? It is present in the spinal cord. Then it is present in the spinal cord. Now we can see the next cells which you are seeing is the pyramidal cells. So we can see the pyramidal cells. This is pyramidal. What is this? This is pyramidal cells. Okay. This is pyramidal cells. So I will write below. Just a minute. And you already know pyramidal cells are present in cerebral cortex. Okay. Now we can also see the enlarged view of flag-shaped cells. Can you see here? This flag-shaped cells which you are seeing, this is the flag-shaped cells, which is Purkinje cell, which is flag-shaped cells. Flag-shaped cells. And if I talk about flag-shaped cell, it is Purkinje. Purkinje cells. It is Purkinje cells. Okay. Now we have done this. Now we are talking about next important points which I want you to tell. So we have categorized the neurons according to the shape, according to the polarity. We have also classified it according to the length of the process. Now the next variety is the functional classification. Neurons can be sensory neurons or the neurons can be motor neurons. So functional classification is functional classification. So till now, my dear students, have you understood? Please let me know. If anything you have not understood, please let me know. I will revise it. Okay. Let me check the messages also. Okay. Okay. Class timing is 6:30. 6:30. It is 6:30 p.m. dear. 6:30 p.m. Okay. It's 6:30 p.m. Now, so we will have two varieties. We will have two varieties which you have to know. One is sensory neuron and the other is motor neuron. Sensory and motor neurons. Okay. So basically you have to know about the sensory neurons and you have to know about the motor neurons. So sensory neurons will receive sensory stimuli from the receptors. Okay. So either they can receive sensory stimuli from the environment or from the within the body, that is from the receptors. So examples are bipolar neurons and pseudo-unipolar neurons. Okay, so let's write about sensory neurons. We have to know about the sensory and we have to know about the motor neurons. It is important that we should be knowing about sensory and we should be knowing about motor neurons. So firstly, we should know about the sensory neurons. So if I talk about sensory neurons receives sensory stimuli. They receive sensory stimuli from environment, that is from receptors, and from within the body. Also, they receive stimuli from the body, from within body. Okay, this will be sensory. And what are the important varieties of sensory neurons? So we will have pseudo-unipolar neurons and bipolar neurons as the example of sensory neurons. Examples. pseudo-unipolar neurons and bipolar neurons. Okay. Now, it is also important for us to know that we should be also, so basically, if I talk about sensory neurons, if I'm talking about sensory neurons, the cell body of all sensory neurons lies outside central nervous system except mesencephalic nucleus of trigeminal nerve. So I just want you to write this note point about sensory neurons. So please write a note point about sensory neurons, note point. Okay, that is an important point. The cell bodies of all sensory neurons lie outside central nervous system, except mesencephalic nucleus of trigeminal, except mesencephalic nucleus of trigeminal nerve. Done? So all these points which I am writing as a note point in between my class, these are very, very important point which you should not leave it. This is very important point which you should not leave it. You should know about these points. Okay, this is highly important for you. So then the motor neurons. Now what about motor neurons? So motor neurons control effector organs. They control muscles. They control exocrine, endocrine glands. So they are located in the central nervous system except that of postganglionic autonomic neurons. Okay, they can be upper motor neurons or they can be low motor neurons. So let's know about, so we have done about the sensory neurons. Now I would like everybody to note down about motor neurons. Okay, so let's also note down about motor neurons. So next variety is motor neurons. Motor neurons. So in functional classification, we have got sensory neurons and we have got motor neurons. So we have done about the sensory neurons. If I talk about motor neurons, the function is it controls effector organs. It controls effector organs. So what are the effector organs which it controls? What are the effector organs which it controls? It controls muscles. It controls exocrine gland and endocrine glands. So they are located, if I am talking about motor neurons, they are located in the central nervous system except postganglionic autonomic neurons. So this is again a note point about the motor neurons. Motor neurons are located in central nervous system except postganglionic neurons of autonomic nervous system. Postganglionic autonomic neurons. Okay. Now, it's also important for you to know about upper motor neurons and lower motor neurons. So, basically, if I am talking about motor neurons, they are of two varieties, upper motor neurons and lower motor neurons. So, basically, motor neurons is of two varieties, upper motor neurons and lower motor neurons. Okay. Now, upper motor neurons are confined to cerebral cortex. They are confined to cerebral cortex. This is an important point. And if I talk about lower motor neurons, so they are confined to cerebral cortex. And if I talk about lower motor neurons, they are confined to anterior horn cell of spinal cord and brainstem. They are confined to anterior horn cells of spinal cord, brainstem. So low motor neurons are present in brainstem and spinal cord. Low motor neurons are present in brainstem and spinal cord. Okay. So done with this. Okay, my dear students. So now, so say here you can see again in this diagram what you can see. Upper motor neurons in the motor cortex of the brain Low motor neurons in the brainstem or in the spinal cord. Low motor neurons are in the brainstem and spinal cord. Okay. Now, let's talk about also about the synapse. So we should know about different types of synapse which happens. So we should be knowing about its classification. So we will have actually the synapse are exodendritic, axosomatic, and axonal. Three types of synapse are there. So let's talk about the next important topic is synapse. So when I'm talking about synapse, it is firstly, before exactly telling about the synapse, so basically it is actually it is important for you to know, so that it's a site of contact which happens between two neurons. So what is, in simple terms, if I want you to know about synapse, it is a site of contact. It is a contact which is happening between the two neurons for the transmission of information, that is called as synapse. Okay, that is what you have to define. It is a specialized junction. Okay, so it is also important for you to know that what are the parts of synapse. So we have to know about the parts of synapse. So we have what presynaptic part, presynaptic, we have got postsynaptic part, and we have got exactly the synapse which is happening. So we have got these three parts: synaptic cleft, we have got postsynaptic membrane, and we have got presynaptic membrane. These are the parts of synapse. So what is synapse, what are the parts of synapse, and then the classification of synapse which you have to know. Okay, so please write down. First thing is, what is synapse? Synapse. So it is the site of contact between two neurons. Between two neurons for the transmission of information. For the transmission of information. Okay. Now, it is actually a specialized junction. When I am talking about synapse, what is synapse? It's a specialized junction. It is specialized junction. And it is important for us to know the parts of synapse. We have to know how many parts the synapse is having. Synapse is having presynaptic membrane, synaptic cleft, and postsynaptic membrane. That is the part. So that is also important for you to know. And then you have to know the different varieties of synapse. So let's write now. Let's focus on this point. So please write down the heading: parts of synapse. Parts of synapse. So we want to define parts of synapse. We have got following three parts of synapse. Okay. So the first is presynaptic membrane. Presynaptic membrane. Okay. So we have to know about this. What is presynaptic membrane? So actually, if I am talking about presynaptic membrane of the presynaptic neuron, so it is a part of presynaptic neuron which contains the neurotransmitter. So presynaptic membrane of presynaptic neuron contains neurotransmitters. Okay, so neurotransmitters. Actually, we will study about these also with the physiology part. Physiology class में भी हम पढ़ेंगे. And the next part is synaptic cleft. Synaptic cleft. So the space which is lying between the pre and post synaptic neuron is called as synaptic cleft. So the space between, if I am talking of the space which lies between pre and post synaptic neuron is called as the synaptic cleft. What is it called as? Synaptic cleft. So this is the area where the neurotransmitters are released. So this is the area where neurotransmitters are released. So this is the area where where neurotransmitters released. Okay. Now, what is the third part? The third part is the postsynaptic membrane. Postsynaptic membrane. Postsynaptic membrane part of postsynaptic neuron. This is the part of postsynaptic neuron where action potential is generated. Where action potential is generated. Done? So we have done here the parts of synapse. Now we are knowing the parts of synapse. There is presynaptic membrane which contains the neurotransmitter. Synaptic cleft is the area between presynaptic and postsynaptic neurons. That cleft is where the neurotransmitter are released, and postsynaptic membrane is where the action potential is generated. In very simple. In details you can see in the physiology session. Now I want you to know about classification of different types of synapse. So 3 types of synaptic classification synapse are there. Dendritic, which is the most common. Then we have got exosomatic, and we have got the least common, that is axonal. So please note down this. So this is an important point. So let's know classification of synapse. Next is to know classification of synapse. Classification of synapse. Okay, so it's important to know the classification of synapse. So actually the most common type of the synapse. So basically the first classification is exodendritic. Exodendritic. This is the exodendritic type. So exodendritic type of synapse is the most common. In this case, so what happens in the exodendritic type, the axon of presynaptic neuron will make contact with the dendrite of the postsynaptic neuron. So there is contact which is taking place between axon and dendrite. Okay, by the name only it is clear exodendritic. So this is the most common type of synapse which happens between the axon of presynaptic neuron and dendrite of postsynaptic neuron. So please write down most common synapse. It is the most common synapse variety. Okay, and in this case of exodendritic synapse, axon of presynaptic neuron make contact with dendrite of postsynaptic neuron. Done? Okay, done. Now coming to the second variety, which is exosomatic. So if you are remembering the name, you can understand. Exosomatic is also less common type. It is also less common. And here the axon terminal of presynaptic neuron come in contact with the cell body of postsynaptic neuron. So in this case, axon of axon terminal will be a better word. Axon terminal of presynaptic neuron comes into contact with cell body of postsynaptic neuron. Okay, got it? Got it, dear? Okay, so we have seen the two varieties. Which is the last variety, my dear students? The last variety is So the last variety, let's talk about the last variety. The last variety is the third variety. This is axonal. This is the least common. This is rare. This is least common. This is very rarely happening and this is least common. The axon of presynaptic neuron will contact with the axon of postsynaptic neuron. So here in this case, the axon of presynaptic neuron contact with axons of postsynaptic neuron. Axons of postsynaptic neuron. Okay, dear. So we have finished this classification also. So we have finished the topic of neurons, but I also want you to tell about neuroglial cells. Okay. So let me just. Okay, ma'am. Clear. CNS kya? Okay. Central nervous system is consist of. Okay. Okay, central nervous system is consist of brain and spinal cord. So basically we are now talking about the general concept to be understood before the neuroanatomy session. So all the topics which I have completed in general anatomy part, that is general information about the joints, general information about the muscles, tissues, cartilage, bones, skin, fascia, general planes of the body and anatomical positions. And today we are talking about the general understanding of general anatomy of nervous system. Okay, so done. Now we have done this. Now the next thing is to know about, so we have finished with the topic of neurons, and the next thing which I want everybody to know is neuroglial cells, the supporting cells of neurons. So let's talk about the supporting cells of neurons. Okay, so done with this, let's talk about neuroglial cells. Okay, so before that, the same thing, as you can see, there is exosomatic. So in this diagram you can see exosomatic, exodendritic, which is most common, axonic synapse. Okay, this is the most common, that is exodendritic, where axon and dendrites are synaptic. This is the axonal synapse, and this is exosomatic synapse, which is also less common. The least common is axonal synapse. Okay, so we have done about the three varieties of synapse. Now we should know about neuroglial cells. So neuroglial cells are actually providing structural support to the neuron. So how we can define neuroglial cells? Neuroglia. So what do you mean by the word glia? The word glia means glue. Okay. So you can see neuroglial cells are providing support to the neurons, and they are acting as a connective tissue framework support structure for the nervous system. So they are actually acting as a connective tissues of nervous system. Okay, they are acting as a connective tissues of nervous system. Now here we can see this are the supporting network. These are few neuroglial cells. Okay, one by one I will describe each neuroglial cells. These are the neuroglial cells. We have to understand what are neuroglial cells. So they are giving structural support to the neurons in the central nervous system. They are also giving support to the peripheral nervous system. So, and they are also capable of mitotic division. So these are the important points about. So we have to know about neuroglial cells providing neuroglial cells provide structural support. They provide structural support for neurons. They provide structural support for neurons, that is in central nervous system. In peripheral nervous system, the other support which it forms is the formation of myelin sheath, which they form, and they are also capable of mitotic division. They are also capable of mitotic division. Now, it is important for you to know the supporting network, the supporting cells, neuroglial cells present in central nervous system, the supporting neuroglial cells which is present in the peripheral nervous system. So, I want everybody to know that. So, please write down. Neuroglial cells in central nervous system. So what are the important neuroglial cells in the central nervous system? And what are the important neuroglial cells in the peripheral nervous system? Neuroglial cells in peripheral nervous system. So this we have to know. Is it okay? So neuroglial cells in central nervous system, important one is it contains astrocytes. Okay. It contains oligodendrocytes. It contains microglia. It contains ependymal cells. Ependymal cells. Now talking about neuroglial cells which is present in the peripheral nervous system, that is Schwann cells, satellite cells, stellate cells, stellate cells, stellate cells, stellate cells. Stellate cells. Okay. So one by one, let's talk about each one of it. So firstly, I would like to talk about astrocytes. So you can see this is how the astrocytes looks. So actually, if I'm talking about astrocytes, it is the cell, it is a cell of the central nervous system, okay? And it is actually providing nutrients to the neurons. What is its function? It is providing nutrients to neurons. This is the function of astrocytes, and it is also very important as it is maintaining blood-brain barrier. So it regulates the blood flow in the brain and maintains synapse also. So it is also forming the basis of blood-brain barrier. And it also provides nutrition to the neurons. So this is the function of astrocytes. So let's write down the function of astrocytes. Astrocytes. So first thing is where astrocytes are present. They are actually, astrocytes are the cells or present in central nervous system. Okay. They support the nervous system. They support central nervous system. They support the nervous system by providing nutrition. They also maintain blood-brain barrier. So they are an integral part of, if I'm talking about astrocyte, food plates of astrocytes, they are integral part of blood-brain barrier. So they are also maintaining the integrity of blood-brain barrier. BBB, triple B, P triple B. What is triple B? They maintain blood-brain barrier. I will show you also the layers of blood-brain barrier. Okay. So this is an important function. Regulate the other important. So this is one of the important function. This is the second important function. If you want to add on more, they also regulate blood flow in brain. So they also regulate blood flow in brain. Okay. Okay. And they also maintain the synapse and maintain the synapse and maintain the synapse and maintain the synapse. Now talking about the next cells. So we have talked about the astrocytes. Now let's talk about oligodendrocytes. The next type of cell is oligodendrocytes. This is commonly asked in examination. Oligodendrocytes are myelinating cells of central nervous system, and this has been several times asked in the examination. So actually oligodendrocytes maintain fast and efficient transport of neuronal communication, and they form myelin sheath in central nervous system, as you can see in this diagram also. Myelin sheath is formed by oligodendrocytes in central nervous system. So please write down about oligodendrocytes. Oligodendrocytes are the myelinating cells of central nervous system, of the central nervous system, okay, that allow allowing fast and efficient transport of neuronal communication. Fast and efficient transport of neuronal communication. So this neuronal communication happens via myelin sheath. Okay. So that is an important point. And actually, if I am talking about this myelin sheath, this is also very important for forming an insulation, electric insulation. And this is important for the fast conduction of the nerve impulses. So this is oligodendrocytes are very important for the fast saltatory axonal conduction. Okay, this is one of the important function of oligodendrocytes. So done with this, now moving on to the next cells, that is microglia cells. So they are small cells, as you can see, they are small and they are small elongate. This will be small cells elongated and will have few processes. As you can see, the microglial cells as having few processes. So let's talk about microglial cells. They are also a type of, so this microglial cells are having, they are small cells. As you can see in this image, they are small cells, can be elongated shape also, elongated body. As you can see, the body is elongated bodies, few processes. Okay, now, so it is actually microglial cells are again a type of microglias are again a type of neuroglial cells which is located in the brain throughout. It is located in the brain. It is also located in the spinal cord, and it is actually if it is actually found in the brain and spinal cord, and they are acting as a macrophage. They are means they are protecting the neurons. So they are actually acting as a microphage cells and act as phagocytizing any invasion. So that is an important function of microglial cells. So please write down microglial cells are present throughout the brain and spinal cord. The brain, it is present throughout or within the brain. So basically it is present in the it is present in the part of central nervous system, present in brain and spinal cord, and they are actually importantly acting as a macrophage cells, which is important to maintain the immune defense. So they are actually phagocytizing the foreign invasion. So they are acting as a immune defense. In central nervous system. Okay. Now talking about next cells. So next of the cells is ependymal cells. Actually ependymal cells, as you can see, this will be the part of central canal. So basically ependymal cells are ciliated columnar cells, and these ciliated columnar cells are lying in the inner lining of the ventricles of the brain and also in the lining of inner aspect of the central canal. So, and basically these are having cilia. They are columnar cells and having cilia. So you can see this is ependymal cells, which is showing you the cilia. So cilia are present in ependymal cells. An important point which it is also asked many times in the MCQ, and also it can, the cilia in high power view can be also appreciated in the histological images. So let's see ependymal cell in the central canal of the brain. So if I talk about ependymal cell, it is present in the central canal of the spinal cord. It is present in the inner lining of the ventricles of the brain. So let's enlarge this. So when I am enlarging this, okay, so you can see the ependymal cell. So you can see this is the central canal of the brain. So this is the central, sorry, this is the central canal of the spinal cord, and you can see inside we have got the ependymal cells, that is columnar cells lining the inner aspect of the central canal of the spinal cord and the ventricles of brain, ciliated columnar cells. Okay, so please write down about ependymal cells. So let's write about ependymal cells. Ependymal cells are present in the inner lining of central canal of spinal cord and central canal of spinal cord and ventricles of brain. ventricles of brain. Lateral ventricle, third ventricle, fourth ventricle. And if I talk about these type of glial cells, it is important for you to note down that they are of the lining epithelium of ependymal cell is ciliated columnar epithelium. Ciliated columnar epithelium. The lining epithelium of the ependymal cells are ciliated columnar epithelium, which you have seen, and they are actually very important part of the central nervous system. They are importantly, why? Because they are also they play an important role in CSF production, and they are flow of CSF. So they maintain the flow of CSF. So it is controlling the production. So it is controlling the formation. It is controlling the formation and flow of CNS. It controls the formation. What is its function? It controls the formation and flow of CSF, cerebrospinal fluid. Cerebrospinal fluids in the central canal of the brain and in the ventricles of brain. This is an important function. So we have done with important neuroglial cells which is present in the central nervous system. Now we have to know other neuroglial cells which are present in the peripheral nervous system, which you have to note down. So let's see this. So let's see this image, which is very, very important for you to understand the Schwann cells. So if I talk about Schwann cells, and that is a very and satellite cells. So two types are there. One is satellite cells and the other is Schwann cells. So in this diagram, we can see the Schwann cells. Okay. So these are satellite cells, and here we can see the Schwann cells. So we have to know that these are the cells, neuroglial cells of peripheral nervous system. Firstly, let me show you. So if I talk about Schwann cells, you can see it forms myelin sheath in peripheral nervous system. So Schwann cells are also called as neurolimocytes, and they are important for the glial cells, which is forming myelin sheath formation for the peripheral nervous system. So they are supporting the neurons. So their function will be giving support to the neurons of peripheral nervous system. Okay. Now the satellite cells. So can you see the satellite cells? So if we are talking about satellite cells, they are also glial cells, as you can see in this diagram. These are the satellite cells. Okay. So these are the Schwann cells and these are the satellite cells which you are seeing. These are the satellite cells. Okay. So basically satellite cells are glial cells, which is actually an important glial cells of the peripheral nervous system, and they are actually very important, and they are found in the sensory, sympathetic and parasympathetic ganglia. So they are also forming a support framework for the neurons of the peripheral nervous system. So let's write about both of it. So let's write about, so firstly I would like to write about Schwann cells. About Schwann cells. So Schwann cells are also called as neurolimocytes. Okay, they are also called as neurolimocytes and are the principal. They are actually the main or the principal glial cells. They are principal glial cells of peripheral nervous system. Okay, and the function is to form myelin sheath. So if I talk about Schwann cells, they form myelin sheath around axon in peripheral nervous system. Okay, glial cells has a function of supporting the, so they support the neuron. Actually, the major function is supporting the neuron, supports the neurons of the peripheral nervous system. Now, let's talk about satellite cells. So next is the satellite cells. And if I talk about satellite glial cells, they are covering the surface of neuronal cell bodies. Satellite neuroglial cells cover the surface of neuron cell bodies in the ganglia of peripheral nervous system. Where they are found? They are found in the ganglia of peripheral nervous system. So basically they are found in ganglias. So they are found in sensory, they are found in sympathetic and parasympathetic ganglia. Okay. Okay, so done. So we have done with this. Now I would also like to discuss one of the important point which everybody should know. So my dear students, are you understanding? Everything is clear? Okay, so that is very, very important for you to note down. So let me just check in 70% paralysis ma'am, body मेरा BSc के class होता है ma'am second year का. Okay, so yes, so let's talk about blood brain barrier. So if I am talking about the blood brain barrier, it is actually important for you to know that it is a selective semi-membrable membrane which is lying at the capillary level, and that is separating. What is the function? It is separating the blood from the brain and allowing the passage of selective contents of blood to that of central nervous system. So that means it is preventing the toxic structures, toxic substance or harmful substance to not pass inside the brain. This is an important function, and it is also important to know how is the blood brain barrier formed. So if I am talking about the blood brain barrier formation, it is formed by the capillary endothelium cells, the basement of the endothelium, and the end feet or food plates or food process of the astrocytes, that is neuroglial cells. So let's see here. This is showing you the blood brain barrier, where you can see these are the astrocytes, that is the neuroglial cells, and these are the blood capillaries. So endothelials of blood capillaries. So endothelial cells, capillary endothelial cells, basement membrane of the capillary endothelial cells, and the end feet of astrocyte together, they form the blood brain barrier, which is restricting the passage of toxic and harmful substance to get inside the brain. That is an important point. So firstly, we should write what is blood brain barrier, what are the constituents or parts of blood brain barrier, and I would also like to enumerate few of the areas where we find no blood brain barriers. Okay, so here in this diagram you can see the food. What is this? This is the food process of blood brain. This is the food process of astrocytes. Food process of astrocytes along with the basement membrane and endothelium of the capillaries, endothelial cells of capillaries, will lead to the formation of blood brain barrier. Okay, that is important for you to note down. Okay, so now let's write it. Everyone, please write down. So write down firstly, what is triple B? What is triple B? Triple B is blood brain barrier. Blood brain barrier. Okay, it is selective semi-permeable membrane at the capillary level that separate the circulating blood to from the brain. So it is separating the circulating blood from entering the brain. Semi-permeable, it is semi-permeable membrane at the capillary end, at the capillary level that separates the circulating blood from brain. Okay, allowing the passage of selective contents from blood to nervous system. This is an important aspect of blood brain barrier. Now, the other important point is, if I am talking about blood brain barrier, it is preventing. So it is preventing the entry of harmful substance or toxic material getting inside the brain. So that is an important point. So basically, if I talk about blood brain barrier, it prevents entry of toxic and harmful. It prevents the entry of toxic and harmful substance from entering brain. This is an important point about blood-brain barrier. This is an important point about blood-brain barrier. Now, it is also important for you to know the constituents and the layers of blood-brain barrier. Then it is important to know which are the areas of the brain where we have got no blood-brain barrier. That means infection chance is more at that location. So please write down. Next, you should write down constituents of blood-brain barrier. So it's also important for my dear students, are you understanding or not? Please let me know. If anything you have not understood, please ask me. So let me just check with the students. Anything which you have not understood, please ask me. Okay, dear? Okay. So let's write the constituent of blood-brain barrier. So it is formed by capillary endothelial cells. It is formed by capillary endothelial cells. It is formed by basement membrane of endothelial cell. It is formed by end feet of neuroglial cell. Which neuroglial cells? Astrocytes. It is formed by end feet of astrocytes covering the capillary wall. Covering the capillary wall. Okay, that is an important point about the constituents of blood-brain barrier. Okay. Now, it's also important for you to know the structures which is not having blood-brain barrier. Okay, so it is also important for you to know the structures which is lying outside the blood-brain barrier. Primarily, the specialized region is called as circumventricular organ. So please write down. So then I will. This is the last and important topic which you should know. So please write on this. Structures lying outside the blood-brain barrier, or structures lying outside. So you have to know the structures which is not lying, which is lying outside the blood-brain barrier. That is together called as CVOs. CVOs means circumventricular organs. Structures outside the blood-brain barrier. Structures outside the blood-brain barriers. These structures are called as CVOs. CVOs. Okay. So what is it? Circumventricular organs. Circumventricular organs. Okay. So basically that means these are the key structures which is the circumventricular organs are following. So this, like for example in neuroanatomy you will see this structure. So we will have one of the structure as area prostremia. Area prostremia is actually vomiting center which is important, which is the circumventricular organ. So first structure is area prostremia, which is vomiting center. Okay. Median eminences. Actually, all these structures you will see in the neuroanatomy class, but I want you to give the list. I want everybody to just note down the list here, but all these structures you will see in the session of neuroanatomy. So median eminences means that is connecting the hypothalamus with the pituitary. So if I talk about median eminences, it connects the hypothalamus with the pituitary. So median eminences connects the hypothalamus to pituitary gland. Okay. So this is an important structures. Also, we will have the third structure, which is called as subfornical organs. Subfornical organs. That is SFO, subfornical organs. Okay. So this is also important for regulating the blood flow and fluid. Subfornical organs, that is important. And okay, other list are, so I'm just completing the list. So I just want you to complete this list of structures which we are talking about. Okay. Done. Yeah. So other than that, we will also have other organs like which is called as the organs close to lamina terminalis. Okay. So vascular organs of lamina terminalis. Vascular organs of lamina terminalis. In short, it is called as OVLT. What is it? OVLT. 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 Also, the other structure like posterior pituitary gland. Posterior pituitary gland in this list. Pineal gland is also in this list. Choroid plexus of the ventricles are also in this list. Okay. So basically, choroid plexus of the ventricles are also in this list. So basically, these are the important structures, which is the structure which is lying outside the blood-brain barrier, which you have to know for your MCQ session. This is very important. Okay. So yes, my dear students, we have completed the topic of neurons. Okay, we have completed the topic of general anatomy of nervous systems, and before ending I would like to just recapitulate what all things you have to know. So today we have discussed in much more detail about the general anatomy of nervous system. So what all things we have covered? Firstly, I have shown you the classification of the nervous system as central nervous system, peripheral nervous system. Then I have told you about the neurons and its structures. I have told you about the cell body, what it contains inside, what all structures are seen, what is Nissl granules, what is axon hillock, what is dendrites. Okay, that all structures we have done. Then we have talked about the classification of neurons according to the polarity. So we have talked about so what all things we have done? We have talked about the unipolar neurons. We have also talked about the pseudo unipolar neurons. We have also talked about pseudo unipolar neurons. We have also talked about the multipolar and bipolar neurons. These are very, very important things which you should not. So just a recapitulation. Can anybody tell me what are neuropolars? Neuropolars examples are unipolar neurons are mesencephalic nucleus of trigeminal nerve. Bipolar neurons are the neurons in the retina, olfactory neurons, and that of neurons of the sensory cochlear nerve and the vestibular nerve. Okay. All these are lying in the list of bipolar neurons. We have also talked about pseudo unipolar, which is appearing unipolar, but the fact is that a single projection process is divided in T-shaped manner and it's pseudo unipolar, and we are knowing the example, dorsal root ganglia. Multipolar neurons, most of the neurons are multipolar. The neurons found in cerebral cortex, the neurons found in cerebellum, the neurons found in spinal cord. After that, we have also discussed about the neurons classification according to the length of processes. That is Golgi type 1 and Golgi type 2 neurons. Thereafter, we have discussed the neurons classification, which was based on the shape. Can anybody tell me the shape neurons, different types of shapes of the neurons and their classification? We have talked about the pyramidal cells present in the cerebral cortex. We have talked about the Purkinje flask-shaped cells present in the cerebellum cortex. We have talked about the stellate cells and all, and we have also talked about, and we have seen various images to identify. Then we have talked about the functional classification of neurons. When I was talking about the functional classification of neurons, I have told you and given the concept of sensory neurons and the motor neurons. I have also told you that the examples there of the sensory and motor neurons, and I have also told you about the concept of upper motor neuron and lower motor neuron, where it is located. Thereafter, we have talked about synapse. Anybody remembers what is synapse? It is the contact which is happening between two neurons for the transmission of information. Okay, so this synapse, in that understanding of synapse, I have also told you about the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. Thereafter, we have talked about the classification of different types of synapse. How many types of synapse are there? Exodendritic synapse, the most common type. Exosomatic synapse, and then exoaxonal synapse, which we have talked. Okay, which is the least common type? The least common type is exoaxonal. My dear students, neuroglial cells is very important for MCQs. We have discussed about different neuroglial cells. I have discussed about all the supporting network of neurons, that is neuroglial cells present in the central nervous system and those who are present in the peripheral nervous system. That also we have discussed. So in the central nervous system, we have got different types of cells, like we have got astrocytes. Can anybody remember the function of astrocytes? Anybody? It maintains the blood-brain barrier. Then we have discussed oligodendrocytes. What is the function of oligodendrocytes? It forms the myelin sheath in the central nervous system. Then we have talked about also the microglial cells, which has got the function of macrophage cells, and it is importantly acting for the immunity to provide to the central nervous system. ये बहुत important है immunity के लिए. And then we have talked about ependymal cells. This is the neuroglial cells of the central nervous system. And where we find the ependymal cell? The ciliated columnar epithelium is found in the inner lining of the central canal of the spinal cord and that of inner lining of the ventricular walls. What is its function? It maintains and secretes CSF, cerebrospinal fluid. Okay, so these were the most important type of neuroglial cells which we have discussed present in central nervous system. Can anybody tell me which type of neuroglial cells were present in the case of peripheral nervous system? In that category, I have explained two neuroglial cells in the peripheral nervous system. What are that? In that category, I have told you about Schwann cells and I have told you about the satellite cells. So Schwann cells is very important neuroglial cells and most important neuroglial cells, which is also called as neurolimocytes, and it is importantly maintaining and forming the myelin sheath in the peripheral nervous system. And the next was satellite cells. So these are also, these two cells are very important neuroglial cells supporting the peripheral nervous system. And at the end, I have shown you an image in which we have discussed about blood-brain barriers. Okay, how the blood-brain barriers are formed, what is the function of blood-brain barrier, and we have also discussed the structures which is lying outside the blood-brain barriers, and in together we called it as circumventricular organs. And I am at the end of the slide where all the structures which is lying outside the blood-brain barrier has been enumerated. Is it okay? So all these structures like area prostrema, that is vomiting center, median eminences, subfornical structures, posterior pituitary gland, pineal gland, lamina terminalis, and choroid plexus. All this list of structures are lying outside the blood-brain barrier. So is it okay? Can we end the session now? Hope the session is useful for you. If anybody is having any doubt, please let me know. Otherwise, I will end the session. And I would like to tell, like tomorrow again, we are meeting at the same time. So I am always meeting with a new session. And so the good point about today's session is that we have come to the end of our general anatomy sessions, or this was the last topic of general anatomy which I have covered, and now I will start from tomorrow the gross anatomy. So please do come timely for the session. 6:30 p.m. I will start, and I will start with the first topic of gross anatomy, upper limb, tomorrow. Okay, see you then. Thank you, all the best, and keep studying. Thank you so much. Okay, so from tomorrow we are going to start. Actually, the whole upcoming session is already on the Prof Essentials. So be the part of Prof Essential Prep Ladder new initiative, that is Prof Essential. Be the part of it. I am there to help you with the subject of anatomy. Thank you, all the best, and keep studying. See you tomorrow at 6:30 p.m. live. Thank you, dear students.